Good afternoon to all, and good evening or good morning to wherever you are calling from. My name is Ben Durham. I am the moderator for today's session, and I would like to welcome you very warmly, and in particular to all the speakers. Just a couple of notes on the moderation rules. We will not be taking questions during the presentations. There is a question and answer session immediately after the last speaker has spoken. And so please jot down your questions for asking then. After each presentation, there will be a short quiz that we are getting your opinion on, and the polls will be shared with us during the question and answer session afterwards.
Okay, we would just like to introduce the chargé from the US Embassy in Pretoria to just deliver those opening remarks via video presentation before we get started on those speaker presentations.
Good afternoon, everybody. I want to welcome you all to today's webinar on genome editing in agriculture, and thank CropLife South Africa for its leadership in organizing this important event. I'd also like to thank the Agricultural Research Council, Biosafety South Africa, and Sansor for its support in bringing about this program as well. In March, I became the U.S. Embassy's Chargé d'Affaires, and I quickly became aware of the strong U.S.-South Africa relationship in terms of agriculture. Last year, despite the challenges posed by COVID-19, our countries achieved more than $800 million in bilateral agricultural trade. It's incredible. In addition to our strong trade ties, the United States and South Africa also have a deep relationship on agricultural research through academic exchanges and scientific support. Today's event highlights the use of science in agriculture and shows how much our countries can achieve when we collaborate. Biotechnology provides farmers with tools that can make production cheaper and more manageable, and new technologies used for breeding staple crops such as maize and soybeans will be instrumental in securing the future of agricultural production and in ensuring there's ample food available for the growing global population. Additionally, advances in agriculture such as genome editing can help us address issues related to climate change. South Africa's adoption of proven scientific approaches to biotechnology have paved the way for its farmers to increase the production of maize exponentially over the past 20 years. And as a result, South Africa has emerged as the continent's leader in biotechnology and a reliable supplier of maize across the region and the world. I encourage all of you to participate actively in today's event, as our robust collaboration now will secure food security in the future and help us to meet the demands of climate change. Thank you very much for your attention.
The first speaker then is Dr. Julian Jafta, who is from South Africa. Dr. Jafta is the Chief Director of Plant Production and Health in the Department of Agriculture, Land Reform and Rural Development, and he has responsibilities amongst others for the development of policy, legislation, strategies, and norms and standards in the plant production and trade in plant goods.
Thank you, Ben, and good afternoon to all the participants. Thank you to CropLife SA for this opportunity. I have deliberately today given my title as the Chief Director responsible for plant production. That is my actual job. But as would become evident through this presentation, it is a bit of a challenge to make a presentation today in my capacity as the chairperson of the Executive Council established in terms of the GMO Act, because the work or work is still in progress in terms of developing a regulated framework for genome editing, and therefore I will briefly outline what is it that Council has been focusing on, where we are at, and possibly what the next steps would be. So I would look at key considerations towards developing a framework. I will look at what is it that we've done to date, and then some justification for regulating genome editing techniques or products under the GMO Act, and then look at an approach that we might follow. Firstly, two of the key considerations was whether or not genome editing techniques fall under the GMO Act, meaning is the GMO Act the appropriate place? Is there scope within the GMO Act to regulate genome editing techniques for products? And then if the answer is then yes, what is it? What kind of risk assessed route would be followed? Would this be exactly the same as we've had it up to now for the first generation GMOs? What is it that we've done up to date? To date, there has been consultation processes with various interest groups. I must point out that this has not been an Executive Council driven process. We are aware that several of our key council members, departments which are represented on the Executive Council, have been engaging with other stakeholders, with other regulatory authorities, but it was not a formal Executive Council process. The Council, however, did request a recommendation from the Advisory Committee as to whether or not these new breeding techniques or genome editing products are regulated under the GMO Act. The Council then considered the advice which came from the Advisory Committee, and then based on that advice, the Advisory Committee was then further requested to develop a framework for risk assessment for genome edited products under the GMO Act. This risk assessment framework is currently before the Executive Council. Now, I must point out the important role that the Advisory Committee is playing.
I must just stay still. Yeah.
It's just that it's not keying correctly.
Sorry.
Oh, I must just point out that the important role of the Advisory Committee. The Advisory Committee has and continues to be the scientists which supports decision making under the GMO Act. So obviously their recommendation is then very important for Council to consider, and Council would then consider other policy initiatives, other policy directions in terms of coming up with a final decision. So when this assignment was given to the Advisory Committee, there was quite a lot of intense work that was undertaken, and we really appreciate this work that has gone in. And yes, admittedly so, there has been quite a lot of backwards and forwards in terms of coming to where we are at this point. Other developments that I'd like to draw your attention to, but which was not necessarily processes driven by the Executive Council, is we're all aware that there was a 2017 report by the Academy of Science of South Africa, which was commissioned by the then Department of Science and Technology. We have also been interacting as individual departments with other regulatory authorities, and a lot of that has been with Argentina, because we have a strong collaboration and a memorandum of understanding with Argentina, specifically around biosafety issues. South Africa has also supported one of the first statements of the like-minded group. This is a group that who were making a case for the science-based risk assessment of LMOs in the EU. And I'm mentioning this specifically here because this group obviously then developed its work further to also include a focus on genome editing. But we have not thus far supported these newer statements, which includes now genome editing. But initially, we did find ourselves aligned to their position with regards to the science-based risk assessment for the EU. Then South Africa also is a member of the WTO, the World Trade Organization, and I think in 2018-19, I think 19, there was a statement from Brazil, Argentina, Chile, Uruguay, and Paraguay at the WTO SPS committee. Their statement focused on the harmonization of regional and international regulations for genome editing products. And here, South Africa expressed its support in this forum for the science-based approach, which was expressed or put forward by this group of countries. And I'm mentioning it again, just to give you some sense that we've obviously given it quite a lot of thought as a country, and as it was appropriate, we have given some hint as to where we are headed in terms of regulating genome editing products, genome edited products. The next question is then it's coming to the crux of the matter, whether or not genome edited products are regulated under the GMO Act. I'd first like to start off by reminding you, and I know a lot of you from the local context would be familiar with this, by just pointing you again to what does the Act say. So the Act, Section 2 of the Act says that this Act shall apply to genetic modification of organisms. And then it also goes further to say it applies to the development, the production, release, use, and application of GMOs. So it gives some sense of what is it that the Act, what is the scope of the domestic legislation. Now, if one then focuses on what is then the definition of a GMO in terms of the domestic legislation, just going to read it. No, a lot of you are familiar with it, but the GMO in terms of the Act means in organisms, the genes or genetic material of which has been modified in a way that does not occur naturally through mating or natural recombination or both. If you look at the definition, modified is qualified to say does not occur naturally through mating or natural recombination or both. And this is what we consider the threshold for regulation. And the threshold for regulation is simply not the variability beyond natural genetic variability as has been expressed in some fora. So in considering the scope of the Act and what we consider, what the Act prescribes in terms of the definition of a GMO, the answer to the question that we've been after is yes. And at this point, is yes, genome edited products, genome editing does resort and will be regulated under the GMO Act. And that's a position that I can confirm is a decision by the Executive Council established in terms of the GMO Act. The following section, the following question now results as focuses on, what is the assessment route that we will follow? Will we follow the same route as the first generation GMOs? Again, the GMO Act aims to achieve the responsible use of technology and minimizing and managing potential risks to human and animal health as well as the environment. The implementation of the existing regulatory framework has always aimed to evolve alongside the experiences gained over the years and to promote bio-innovation. We've considered two possible, or we are in the process of, or we have considered two possible approaches in developing the draft risk assessment framework for genome editing products. Firstly, a possible approach would be to use the current system for assessing GMOs with minor modification, or to follow a tiered approach. And in such a tiered approach, information requirements would be clearly stipulated to guide the applicant through the process, but the initial assessment must and would still remain appropriate. So it can't just be a quick slapdash approach. The initial risk assessment must still achieve the objectives of the Act by ensuring that we minimize and manage any potential risk. Now, similar
if you can just point out we have 2 minutes left.
Yeah, I'm almost there, Ben.
Thank you.
Now, similar to first generation GMOs, the potential risk associated with genome edited products would obviously be influenced by the host organisms, the intended change, the receiving environment, as well as the intended scale of use. And these are the issues that would be addressed in the tiered approach. In tier 1, there would be we would need information to conduct a basic risk assessment, and it's those areas that I have indicated, like that we are normally familiar with, but there would also be specific sections for specific GMOs or specific organisms, and this would also make the navigation through the first tier easier for applicants, such that it would not be necessary for them to look at areas where which would not be applicable to their specific activity. Then tier 2 would be supplementary information, and supplementary information would now be those areas that you would be directed to based on the responses provided in tier 1, and it may include info such as unintended effects, info on gene drives, human and animal health issues. Then the tiered approach we envisage would ultimately also cover first generation GMs, but we also then still need to ensure that the information that we would be requiring for submission still needs to comply with Annex 3 of the Cartagena Protocol. And then to do that, we will have to still adjust some of our administrative process. To date, we've not had any formal application under the GMO Act for a genome edited product, but we've had an inquiry on genome edited tomatoes, and as to how it would be regulated. The next steps, this is the last slide, Ben. The next steps would be consensus approval of the risk assessment framework by the council, and then if there are any guidelines which we expect would be required, that to support the risk assessment framework, that these would need to be approved by the Minister in line with Section 5(2)(f) of the Act, and then, if required, the development of additional regulations to support the inclusion or this new focus under the GMO Act. We are aware that consultation has been a major question as to how we would deal with consultation. We don't, unless there are regulations that needs to be developed. Legally, there would be no requirement for any formal consultation. But we will also, as we present this new development to the minister responsible for the legislation, she may then decide how she would want to take forward issues around consultation. I thank you for your attention, and that's the end of the presentation. Thank you. Thank you, Ben.
Right. Julian, thank you so much. Really informative and interesting, and delivered in a calm, measured tones which belie the intense discussion that is happening behind the scenes. Thanks very much for that. Folks, what we're going to do now, as I said earlier, is go into an opinion poll. So if the organizers can display that, please indicate your choice in this, the opinion poll, which you should be able to see now. Alignment of regulatory processes governing genome edited products is essential to what? Please indicate your choice, and we will then give the poll at the end during the question and answer session. If you can please then enter what you believe is the most appropriate response to that question, one of 4 that will be coming through to you. So if I can then introduce the next speaker in this program. The next speaker is Dr. Rufus Ebegba, who is the Director General, Chief Executive Officer of the National Biosafety Management of South Africa. Nigeria, very important. Thanks very much for joining us, Dr. Ebegba. He has responsibilities for the provision of the regulatory framework, institutional and administrative mechanisms for safety measures in the application of modern biotechnology in Nigeria. The talk by Dr. Ebegba will focus on Nigeria's regulatory approach.
Good morning, everyone. My name is Dr. Rufus Ebegba. As said, I'm the Director General of the National Biosafety Management Agency of Nigeria. And I'm going to share my slides. The title of my presentation is Gene Editing in Agriculture, Nigeria's Regulatory Approach. The outline of my presentation includes brief introduction, brief of Nigeria regulatory body, gene editing in Nigeria, rationale behind adopted approach for the gene editing regulation, guideline development process, provision for applications, challenges, monitoring activities, and conclusion. Introduction. The groundbreaking effects of gene editing has been proven on an array of agricultural organisms. Few of genome editing applications have been in the area of introduction of new methods for the management of pests the improvement of the efficiency of agrobias, and attempt to solve diseases in the area of regulation of products. Gene editing, which is a modification of the genome within a living cell, either by the insertion, deletion, or replacing one or more segments of DNA, is now much easier, faster, cheaper, and more versatile than ever. having editing techniques that could allow for new editing applications ranging from viruses and bacteria to animal, plants, and human beings. This fast rising employment of gene editing, deployment of gene editing, however, raises some biosafety concerns. As with the technology, irrespective of its tremendous benefits, the need for it to be properly regulated so that it will not be abused or healthy products can only be delivered is very expedient. Brief of Nigerian regulatory body. The National Biosafety Management Agency of Nigeria was established in 2015. I am the current Director General of the agency and the pioneer Director General of the agency. The mandate of the agency is to ensure with responsibility for providing regulatory framework, institutional and administrative mechanism for safety measures in the application of modern biotechnology and the use and handling of genetically modified organisms in Nigeria. What is the vision of the agency? The vision of the agency is to ensure that the practice, processes, and procedures of modern biotechnology application are undertaken within the limits of regulatory system to guarantee the safety of use and the protection of the Nigerian biodiversity from potential adverse impacts from genetically modified organisms, taking into account risks to human health. The mission is to promote the basic tenets of biosafety as instituted in the Cartagena Protocol on Biosafety and enforce Nigerian National Biosafety Management Agency Act 2015 as amended. The government of Nigeria in 2019 amended the existing National Biosafety Management Agency Act of 2015 to include the regulation of gene editing, emerging aspect of modern biotechnology in the area of synthetic biology, gene editing, gene drive, and also a new aspect of a regulatory system which was all included in the area of biosecurity. However, this topic is basically on gene editing, which guidelines have been developed for. What is the Nigerian biosafety policy approach in the area of gene editing? The National Biosafety Management Agency Act of 2015 as amended. Section 25A states, no person, institution, and body shall carry out gene drive, gene editing, and synthetic biology except with the approval of the National Biosafety Management Agency. The regulation of gene editing in Nigeria is as well involved the containment in the national, they are all contained in the National Biosafety Guideline on gene editing, which has been recently developed and also adopted to ensure that there's guidance in the area of gene editing. And this encompasses ways of which are the gene edited products should be fully regulated and the ones that are not to be fully regulated. In the approach, if a genetic material, a genetically modified organism is edited, or an approach of insertion of a novel gene into an organism, and at the end of the product, if the gene of insert or the foreign gene does not appear in the final product, the process is not fully regulated. And the applicant, if an applicant is informed, regulatory framework of genetically modified organisms. If the gene edited product does not involve the use of any transgene, such product as well does not involve full regulation of regulatory process. But if the final product contains transgene or foreign material that has been inserted, the full regulatory process is carried out. Risk assessment and risk management dossiers are required for it to be fully regulated using the normal biosafety regulation approach. In Nigeria, the gene editing process, as earlier on explained, is very, very simple. Products that contain the transgene, the ones that were used, we use material from another organism to modify but does not have the final transgene, and the one that do not only require insertion of any gene. If it does not require insertion of any gene, it's not taken as a GMO. But if the final product contains transgene, it's taken as a GMO, and if a full regulatory process is carried out. This is the process map, as earlier on enumerated. One, the applicant submits an application to the National Biosafety Management Agency. There is an internal review process, preliminary review process to check whether this product really contains, what is the process that has been used to gene edit the product. If it contains the genetic material, the final product is taken as GMO and full regulatory approach is carried out. If it is, there's a mediated gene and it doesn't, and the final gene the gene does not appear in the final product, it's also not considered as a GMO. In such a situation, the applicant is responded to that this product does not fall within the GMO product, and as such, an abridged regulatory process is applied. But if it contains the final gene, in that case, it is a full GMO and the full regulatory process of the biosafety system in Nigeria applies. But if it is done in a way that no gene was transferred in any form, it's also taken that it does not fall within GMO regulatory process of Nigeria. The rationale behind the policy is to ensure the conscience in the science of genetic engineering is holistic and safe to human health and to the environment, and also the issue of socioeconomic concern also properly addressed. And the product is also held, and it does not lead to the transformation of any new toxin to the product. Policy approach. One, research and discussion within the international, the development of the policy, the guidelines which Nigeria has actually used, took the following procedures. One, discussions to look at the preliminary elements. These discussions were based on international and local interactions. Then two, the formation of an internal committee to draft the regulations. And after the drafting, the draft zero guidelines are subjected to various stakeholders review sessions. This also includes international reviewers for us to have a very good guidelines that meet international requirements. After that, the guidelines were now adopted through a validation by stakeholders. And right now we are at this implementation stage, which is the fourth stage, so that we also test the workability of the guidelines and to know whether this is what we really need. For now, it's going to be used. However, there has not been any application for the submission of any application for approval that requires gene-edited products. Then what are the procedures that is required by anybody that wants to submit an application for approval for a gene-edited product in Nigeria? One is the submission of an application to the National Biosafety Management Agency in the form of prescribed form, then acknowledgement of the application that has been filed in by an applicant, then checking for completeness and to ensure that this product, if it is genetically modified, if fall within genetic engineering products like GMOs, that if you fall a full process is carried out, if it does not fall within that process, that is the preliminary checklisting. It means the applicant, the process will terminate at the third level by communicating the applicant that your application does not fall within the regulation of GMOs. To now enable the applicant to move on with his processes or application to another organization that may require approval before such products are released. The request for additional information in the event that more information is needed, that is also done. The convenience of the decision of the internal review process, earlier on said, will also be sent to the applicant to see when is your application which won't require further biosafety processing, but the product does not fall within the GMO consideration. But however, if it contains the gene of insert, full regulatory process is carried out. Then the payment, in that case, the applicant will have to pay a prescribed fee to the National Biosafety Management Agency. Then other regulatory processes like the publication to in the national dailies for stakeholders to take a look at the application, to make come up with their input with National Biosafety Management Agency to guide the agency in its decision-making process. While that is also going on, the agency establishes the National Biosafety Sub-Technical Committee and National Biosafety Committee to in-depthly look at these two independent bodies to look at the application dossier to advise the National Biosafety Management Agency whether should grant application or not.
Doctor,
then
you have 1 minute left.
Yeah. Then the decision, once it's confirmed that the product is safe, and a decision document is prepared, and also authorization certificate is issued to the applicant. These are some of the session of the review of applications in Nigeria. Then what are the challenges? The challenges in the development of the guideline include identifying the right experts, which were very much not available. Then the financial implication in the conveyance of meetings, and also the socioeconomic consideration in the area whether these products do not meet up our national socioeconomic requirement or otherwise. And also the activists who have constantly fought the agency in the area of granting of approvals. And the final process is the monitoring of the activities of the approval at the end of granting of the approval to ensure that in conclusion. Finally, the National Biosafety Management Agency can impose additional guide and additional terms if risks are discovered to involve the management, and also such permits can also be suspended or revoked, and necessary actions can also be taken. I want to say thank you for listening.
Dr. Begba, thank you very much. Really interesting overview of regulation of genome editing in Nigeria. As I said earlier, please jot down your questions to ask in the question and answer session afterwards, or alternatively write them in the Q&A section at the bottom of your screen. So we will have a look at the next poll now, if you can please display it. The next question is, should genome edited products be regulated differently or the same as GMOs? If you can please put your answer in there. Just 2 options here, so it should be quick and easy. And then we will proceed on to the next speaker. The next speaker then is Professor Alejandro Hernandez, who is the Regional Director of Biotechnology for CropLife Latin America, based in Costa Rica. With that brief introduction, can I hand over to you, Alejandro?
Thank you. I hope you can hear me now.
Yes, we can.
So I can start. So first of all, thank you. Thank you for this space. My name is Alejandro Hernandez. I work as a researcher in genome editing in yeast and in rice in Costa Rica, and I'm currently the director of Central America for CropLife. And I worked as a regulator for almost a decade, and I've been teaching for almost 15 years. And part of my research and work was at the university, and in this university we have this study, and a part of the work was to provide legal understanding of genome editing plants and the development of rice as well. So we helped the Minister of Central America, the Minister of Guatemala and Honduras in the development of its norm with the help of AICA. So basically, if I help you with the legal global status of genome editing, if you can see, you can easily note that Latin America is moving forward in recognizing genome editing products as a breeding tool. And most of the products are recognized as non-GMO, which is quite different from Europe. And so if you see the context of South America and Central America, what you will find is a declaration at the WTO looking forward for a science-based position on this. And basically that was the same by the South America Ministers of Agriculture, and South Africa was also part of this declaration. So why Central America and South America is not considering the EU approach that was triggered by some judge decision that requested to regulate all mutations as GMOs, right? So why are we not considering that approach? Is basically because there is an impossibility from a technical point of view to distinguish the products in the market. So basically, you cannot distinguish human intervention from some spontaneous mutations. I don't mean that you cannot detect all mutations. You can do that, but there is an impossibility to recognize the origin of that mutation. So if you take a look at the decision of the court, Mr. Bolbeck says basically that genome editing products, some of them could be exempted from being a GMO as long as they do not contain DNA from other species. And basically, the new study of the EU goes in the same direction. Now, for countries like our countries, Argentina, Colombia, Brazil, United States, a variety is equivalent to a conventional product when you don't have a foreign DNA, in the absence of a foreign DNA. Now, the rationale for that is that you cannot distinguish the product once in the market. And the problem for that is that you cannot stop, destroy, or manage something that you cannot detect in the market, because you'll have a lot of problems from a liability point of view. If you stop a grain shipment on the basis of a supposition, then you'll have a problem from a legal point of view in our countries. So the rationale is very simple. As long as you don't have anything new, something that you cannot obtain from conventional breeding or mutation, you'll not be able to regulate it differently because you cannot distinguish those in the market. So and if you think in other technologies, that is kind of a default if you try to regulate something that you cannot control. And it's called technological default because you cannot use it because you regulate it, but you cannot detect it because you cannot control it, right? So basically, what we're doing from a legal point of view is that we're using the international framework to understand, to leverage our local definitions of what a GMO is. And if you use the Cartagena Protocol as a basis, you'll have a composition of two parts in the definition of what a GMO is a living modified that possesses a novel combination of genetical material that is obtained through the use of modern biotechnology. And the key word here is novel combination of genetical material. And what we've done in Central America and in South America as well is to provide a clarification of what is the meaning of those words. What is the meaning of novel combination of genetical material, which is what Rufus was saying. You have a novel combination of genetical material when you have a piece of DNA that you cannot obtain through conventional breeding or are not found in nature or provided by mutation. That is basically the way we are defining a novel combination of genetical material. So if you have a piece of DNA that you can detect, that you can have something that you cannot obtain by conventional breeding, then you are in the presence of a GMO under the umbrella of the Cartagena Protocol, under the specific definition of what novel combination of genetical material is. And we're doing this at a Central America level. So this norm, this RT 65060118, is a Central America, is a regional definition, is an international definition that we're using to provide that's the legal rationale for us, right? This is a piece of DNA that you cannot obtain through conventional breeding or not found in nature. And basically what we do is that we have a board of science people that are looking into the pieces of mutations, if you can achieve this by conventional breeding or mutation or a spontaneous mutation. And if you have something new, then it's regulated under the umbrella of the GMO and you provide a risk assessment and all that. If not, then you regulate it as a conventional product. And we're doing that with the help of AICA. AICA is a board of is an America institute that is provided. The board of this institute is the Ministers of Agriculture of the region. And so basically, we define what novel combination is under the international legal definition of the Cartagena Protocol, because at a legal level, it has a constitutional big meaning for us, right? And basically in Honduras, we have the procedure that comes out of this international definition, which is basically a consultation procedure. We review the technical information, and we analyze also the definitions and analyze the technical review done by other countries. So we don't provide a barrier at the international trade based on the WTO declarations that was done by Honduras and Guatemala and Central America. And we have this board of experts that goes step by step, and it does the same thing in Guatemala. We have a procedure that goes through the National Biosafety Committee. It reviews and it makes a declaration. And what we see is that with all of the tools of genome editing, because we're not just talking of CRISPR, but a lot of other tools, we look for a novel combination of genetic material. If you have a piece of DNA that you can detect and you cannot obtain by conventional breeding mutation or natural mutation. So basically that's it. You can review the information that I'm telling you, but if you go through the legal norms in Brazil, Chile, Honduras, and Argentina, is the same approach. We're looking for a foreign DNA in Colombia, which is the piece of DNA that I'm talking. We're also looking for in Chile, there is a big definition, but it's the same. Is a piece of DNA that you can obtain by conventional breeding. It's the same in Brazil. They call it recombination from external DNA. It's the same in Argentina. We call that construct, but is a foreign DNA. And in Africa, we had this meeting in 2019, and we gathered a lot of countries, and we came back. We after a lot of reviews and discussions and come back and forward, we get to the same conclusion. The final product cannot be a GMO when it's similar to what is obtained by conventional breeding, because you cannot regulate that in the market, right? Even though you perform a risk assessment, you do the analysis, once in the market, if you try to differentiate something that you cannot differentiate from a technical point of view, then it's a default, and you're actually you're in problems or in a challenge from a government. And we had these discussions in Malawi, and I think it's something, the definition is similar to what you have in South Africa. And we were discussing about that genetically modified organism is something that does not occur naturally through mating or natural recombination. And the problem is that the healing of the DNA is natural, right? And the way the DNA heals itself, it's called recombination. So we call, when you hit the DNA, when you cut the DNA with CRISPR or any other talents or whatever, you are inducing the natural recombination process. So that's a tricky part, because if you take this word literally, then you can end up regulating GMOs as non-GMOs, right? Because you can have natural recombination and introduce a piece of DNA, a foreign piece of DNA, that for us is a GMO under the Cartagena Protocol umbrella. So what I was telling to Malawi is that perhaps you need to take a look to the national and the international legislation, so you can provide clarity in both directions. So you can comply with all of them, right? So for us, that's it. It's always a pleasure to collaborate. Just to let you know that for those that are not into this topic, when you are using CRISPR or talents or any other technologies, the thing is that you can result in a GMO or in a common mutation, right? And for us, a GMO is when you have a piece of DNA, an external piece of DNA that you cannot achieve by other ways. Now, this is tricky also, because in nature, you can find some pieces of DNA moving through the genome, for example, the transposons. But if you have those, or if you have an allele substitution, which is something that also occurs in nature, then you're in the presence of something that is natural, right? Something that you can achieve by conventional breeding. So for us in Central America and in South America, the keyword, the trigger of the norms has to deal with the piece of DNA that you add, and you cannot obtain by other ways, right? And that's it. And it's always a pleasure to collaborate. And I think that I'm in my in my timing.
You are indeed, Alejandro. Thank you very much. Very informative talk, and emphasizing the importance, amongst other things, of definitions and the wording of the regulations. Really informative. Thank you so much. We will then move on to our next opinion poll, which is the question will be put up on your screens now. Does genome editing have a role to play in improving the resilience and sustainability of farming and the food system? Your opinion on these and the answers to the polls will be put up in the question and answer session. Another quite simple one. So while you're doing that, I will carry on with the introduction of our fourth and last speaker. Dr. Donald Mackenzie is the Executive Director of the Institute for International Crop Improvement at the Donald Danforth Plant Science Center in the US. Donald manages the IICI's programs and partnerships dedicated to delivering precision genetics technologies to meet the significant food and nutritional security challenges faced by smallholder farmers everywhere. He is an international expert in regulatory systems for agriculture and has got partnerships in various places around the world, and has previously worked with the Canadian federal government, the biotech industry, and in the public sector research organizations. Over to you, Donald.
Thank you, Ben. And greetings, everyone, from the Danforth Plant Science Center here in St. Louis. Just by way of background, we are a private, not-for-profit research institute established in 1998 with a mission to harness plant science innovation for the benefit of society. As Ben mentioned, the institute that I lead is involved mainly in doing translational research. That's moving innovation from the stage of proof of concept to products that can be used by farmers and consumers. All of our collaborations are with institutions, both members of the CG, IAR system, and the NARS. Our projects focus on improving pest and disease resistance, looking at improved tolerance to abiotic stresses, to improved nutritional content, and the like in staple crops. We use a variety of technologies. Some of these projects use genetic engineering. Other ones use genome editing. So, for example, the project that we're involved with and collaborating with Heinrich Heine University in Dusseldorf is using genome editing to introduce bacterial blight resistance into rice. Within our own institution, Dr. Getu Daguma is using genome editing to target the green revolution genes in teff. Teff is an ancient grain, a staple food for more than 100 million people in Ethiopia. However, it's severely impacted by lodging, yield reductions of 25% or more. So what Getu and the team are doing is using genome editing to very precisely target and introduce the same kind of mutations in the semi-dwarfing genes in teff that were found by chance back in the middle of the 1960s, occurring in a rice cultivar from China that ultimately became apparent to IR8, which essentially was the basis of the green revolution in rice. So using genome editing, we're able to introduce exactly the same kind of a mutation, but in a much more precise and targeted manner. So it's worth pointing out that all of our conversations and thinking about, plant breeding and that and genome editing have to be taken in the context of looking at the evolution of plant breeding innovations over decades and even longer in that, and realizing that although genome editing may seem like it's the latest, greatest thing, and it is at the moment, it's not the end of the story. There'll be newer and even better techniques in the future. So, and also as Alejandro was mentioning, the vast majority of the innovations that we're creating through genome editing are simple knockout type mutations, the same kinds of mutations that we could get by chance using mutation breeding, for example. That's been around since the 1930s or so. So there's a lot of talk by folks that are concerned about, the unintended effects that might arise from different forms or different plant breeding technologies, or the off-target effects, if you will. And it's useful to point out and remember that all plant breeding methods have some potential to introduce off-target effects. And if we look at mutation breeding, which has been around for 80 or more years, it's all about off-target effects. There's nothing targeted about using gamma rays or using mutagenic chemicals to introduce mutations or chaos in the genome. All we're doing afterwards is looking for those changes that might be beneficial. And for the most part, almost all of these techniques we've been able to manage quite successfully just through our traditional agricultural and food regulatory frameworks. So regulated as conventional, if you will. One of the lessons, I guess, or one of the observations, and it should not be news to most people, is that when we think about, the first generation of genetically engineered plant products, and that the public sector and small and medium-sized enterprises have been largely excluded. And they've been excluded because of barriers to market entry posed by strict regulations, by very onerous, in some cases, information and data requirements. At the moment, at least in the genome editing space, we see a much different mix of organizations involved. This is not a space that's dominated by multinational corporations. It's really largely made up by small and medium-sized companies, by universities, other nonprofit organizations, or government research institutes and that. Coming out of all of this experience and from many of the actors playing in the genome editing space are increasing calls for a more product-oriented focus to regulation. So not regulations based on a particular process, if you will, because we can never keep up with the processes. There'll always be new technologies just around the corner. And any regulations that try and regulate based on the technology will always be playing catch up. So it's really about thinking about the products. And this isn't anything new. We've had, national academies of science and learned bodies saying since at least 1987 that it's all about the product. It's not about the process that you used to make it. Risks are related to the product. And this has been one of the things that we've been hearing from our science academies over the decades, and yet almost without exception. All of our countries in that have chosen to go down the road of regulating based on the process, based on the technique that was used. I just want to use this to make the point that there's no such thing as unregulated. So even for products of conventional breeding and that we're all very familiar with, we have this very elaborate and interconnected system of legislations, regulations, international standards, and that cover every aspect from the production of new varieties and varietal registration and demonstrating that varieties are both efficacious and that they're safe and that they're going to add value, all the way through seed production and grain production and handling. to the end user, to the consumer. So whether you like to say from farm to fork or from gate to plate, we have this elaborate system that's already in place that involves both agencies and departments at a national level. It involves international players in that. It involves international standards. And over decades, it has served its purpose, being fit for purpose to be able to provide assurance that the food supply is both safe and nourishing. So when we think about, how to regulate new products and products of genome editing, we have to remember that we already have this existing system in place. And it's useful to also point out that, some countries have decided that for them, as they think about the products of biotechnology, that they have decided that they don't need to create new authorities. That in fact, there are existing authorities that they can utilize, existing procedures that maybe only need to be amended or adapted to take into account products of biotechnology. So the final is that even, if gene-edited products are exempted or excluded from GMO regulation, they're not unregulated at all. Just a simple illustration of a typical workflow in doing genome editing, where you're starting with designing your guide RNAs and deciding what genes you're going to target, and doing the transformation and regeneration and selection of lines. And then eventually you get to a point of segregating out the gene editing reagents or the transgenes that were introduced temporarily. And then you go into open field testing and varietal performance testing and the like. In a lot of countries, that work that's going on in the lab, in the greenhouse, is regulated under recombinant DNA research regulations and that. And so I think what's important is that as we make the transition from that regulated recombinant DNA work that's going on in the lab to field testing with a final edited line that's free of all of its transgenes, that crossing that, making that transition, if you will, from a regulated activity to a non-regulated product, it's important that the methods that we choose to demonstrate the lack of transgene integration need to be practical. They need to be fit for purpose. We need to be able to demonstrate that before we even go into the field, so that we shouldn't be going into a field trial situation with a genome edited product and treating it as if it was a GMO. And it's also important that systems be set up to account for foreign developed products that may want to be coming into the system at the point of going directly into open field evaluation and trials, provided they can meet the test of being free of introduced transgenes and the like. So just a couple of final thoughts. Food security statistics have been going in the wrong direction for the past few years. Climate change is having an impact. It is real. It is impacting crop productivity, and even some would say impacting the nutrient content of crops. We need to increase crop production by about 50% between now and 2050. So, the challenges are great. Time is short. A typical breeding cycle is about 6 to 8 years if we're talking about a conventionally bred product. We know it's double that if we're talking about a genetically engineered product. So we don't really have the time. And we need to be able to avail ourselves of every technology if we have any hope of achieving the goals. We need to follow the science and capture the learnings of more than 3 decades of regulating GMOs. By now we know, and we've done all of the experiments and funded all of the studies globally to show that, genetic engineering in that is inherently a safe technology. We have not yet found evidence of harm. But we have made the systems so complicated and so costly that we have, in a sense, in many cases, stifled innovation, especially from the public sector. The innovations that need to happen in smallholder crops and food security crops, and that are not going to come from big ag. They're going to come from academia. They're going to come from public sector researchers. We need to make sure that we have systems in place that provide them a path to the farmer's field and that. So I think, when we think about most of the plant breeding innovations that are coming from genome editing now, which are by and large knockout type mutations that could be created in other ways, they're probably more suited to being regulated as conventional than they are to being regulated as GMOs. And my final thought, and this is just an invitation to everybody to come and visit St. Louis, to come and visit the Danforth Plant Science Center, but even more importantly than that, to come to St. Louis next year in April of 2022 to attend the 16th International Symposium on Biosafety Research. Dr. Henny Grunwald from Biosafety South Africa is leading the team that's developing, I think, a very compelling program of topics and speakers. So looking forward to seeing many of you in St. Louis next April. Thanks very much.
Donald, thank you so much. Very interesting overview of what is happening and the importance of the technology, and noting that products are regulated whether or not there is a specific regulation on modification, and that time is short. We are now going to be entering into our question and answer session, but before we do that, we're going to have the last poll question for you. If we can please display that on the screen. The last question is, who is responsible for communication to promote acceptance of genome edited products? And it looks like they have specifically excluded all of the above, so there is no cop-out here. You do need to pick one or the other, although I do notice there is an other. Please, can you do that as I lead into our question and answer session? You will have heard the 4 speakers, some really interesting overviews of what is happening both nationally and across borders in regions and around the world. And this is your chance then to engage with these experts to get some kind of insight into what's happening. The first question then that I would like to pose comes from the Q&A chat box, and it's the very last question, and I see Alejandro is already typing an answer, but can we get a response perhaps from Alejandro to the question verbally? The question is, can you kindly, it's just gone to the answered section, can you kindly in brief differentiate gene editing and genome genetically modified organisms or genetic engineering in simpler terms? Thank you.
But I
so
can.
you have written a response. Please go ahead.
Yeah, just for you. For us, you have a GMO when you have a piece of DNA that you cannot obtain by other ways. I mean, it doesn't matter if you use CRISPR, if you use talents, if you use any other technique. If you have a piece of DNA, a foreign DNA, a piece of DNA that you cannot obtain by conventional breeding or natural mutation or conventional methods, then you have a GMO, because you can obtain a GMO by CRISPR. when you include a piece of DNA that is not there, when you change a promoter in a way that does not occur naturally, then you can result in a GMO from our point of view. Thank you. And that's the difference. It's not based on the technique. It's based on the product that goes to the market, and you can or not differentiate that. The issue for us is that if you are the government and you need to differentiate something, and I was given the example of a scar. When you cut your skin and you have a scar, then you can recognize the scar, but you cannot recognize the origin of that scar. But if you have a tattoo that is something different, that is something new, you can then recognize the origin of that tattoo, the ink of that tattoo, who's the author of that tattoo, right? And you have a piece of information that is new, then you have a GMO, in simple words. Thank you.
Excellent. Does any of the other panelists want to add to that clarification? Otherwise, we will move on. I have jotted down a number of questions myself, but I'm still going to try and give a focus emphasis to the written ones in the Q&A. So folks, if you do have questions of clarity or questions about the interpretation or questions about the meaning and where does it go to from here, please do write your questions in the chat box. The next question I want to engage with is from Wilbert Mutezo, who says, why can't we have a simple international standard on the regulation of gene-edited products? We heard Alejandro talk about the fact that you, in some cases, cannot distinguish the origin of a mutation, and therefore, in some cases, you may not be able to control it. So it is critical that we do have a common understanding around the globe for products that may not be identifiable as genome-edited products. Alejandro, can you give a first stab as why don't we have simple international standards?
Yeah, that's the way we're thinking in Central America. And so that's why we have a Central America umbrella, because we're thinking in local products, not just in international products, but in local products. We are developing, in my case, some rice that we need for us. And the thing is that the technology is available. As Donald said, we can use these tools for local products. So we're looking at this as a sustainable solution for us. And I think that the umbrella and the way to do that is to work together. And that's why we have a Central America umbrella working with South America ministers as well. And I think that should be the right way to address these issues. So we don't get into international trade problems as assigned in the WTO by different countries, including in South Africa in 2019. So I think that's the right way. The correct way to do that is to understand and to go to a simple and common approach. And so we don't get into these issues that, I'm having a banana that for us is not a GMO, but for you it is, but you cannot demonstrate that it's a GMO. So we have some trade issues that should not exist, right?
Thanks, Alejandro. Donald, I wonder if I can follow up with you on that same question, noting that the EU bloc and New Zealand, possibly amongst others, have said they are going to be treating genome-edited products in the same way as GMOs, whereas there are a whole range of other countries, including Latin and North America. And the example that we have is Nigeria, which are saying that, no, we want to treat genome-edited products differently. Is there realistic scope of getting to a common global standard?
Probably not. And I think the reason I say that is that it's not just a science and a technical issue. On areas that are, for example, related to a risk and safety assessment, which are really more about science, and that we do have international standards. We could point to the Codex Alimentarius standard around doing a food safety assessment from products of recombinant DNA research in plants. But the regulation of GMOs or genome editing is also very politically charged. And I think it becomes very, very difficult to align vastly disparate ideologies to come up with a common basis, if you will. So I think the better approach, and it's one that Alejandro mentioned, is really to think about sort of regional or sub-regional blocks, trading partners and neighbors who then may be able to work more closely together to align themselves. Yeah.
Excellent point. Thanks very much, Donald. Julian, your thoughts on this harmonization alignment issue?
Yes, thank you, Ben. I want to wholeheartedly agree with Donald. I do believe that risk perception is very different if in different territories. If one considers if you look at just the agenda of the Sanitary and Phytosanitary Committee at the WTO, a lot of the issues and the areas of disagreement between trading partners or parties relates to the interpretation of standards, and it relates to safety standards. So even in that is already an indication of how beyond the science there are these differences. So the standard setting bodies do participate and they give guidance, but even the very same parties who had agreed to those safety standards, as and when it suits their let's call it their trade or their economic agenda, then those standards becomes irrelevant because there's another agenda unfortunately beyond science. Thank you.
Great. Thanks very much, Julian. Rufus, if I can put a question to you. You gave us an overview of how genome-edited products will be regulated under the Nigerian system. But I understand the draft regulations are still under preparation. Do you have a timeline by when you will be able to consider genome-edited products? Can you give an idea of that?
Thank you very much. The guidelines have actually been validated. They are currently in use. They've been approved by the government. So the guidelines are available to be used. They're already in our website. I think guidelines are very, very clear, very precise. Talked about all because
we seem to have a technical glitch with Rufus from Nigeria.
The law, the amendment to the National Biosafety Management Agency of 20, all gene-edited products will be regulated. So streamline which ones require in-depth regulations and the ones that will be exempted. The ones that do not contain foreign DNA will not be fully regulated, but a clearance will be issued. But the ones that contain foreign gene will undergo the true risk assessment process and be treated like GMO and looked at holistically. That's what we have in our guidelines.
Thank you. But you said, I think, that you haven't had an application received, but just to say thank you very much for showing leadership on the African continent. There has been a fair bit of discussion about which country would come first. Kenya has also been working very hard on its genome editing regulations. But I understand you have put them to the post. A question then from the Q&A section addressed to Julian in South Africa. Please, can you just clarify, using the definition of modified as used by Dr. Jafta, should we not also classify and regulate products developed via random mutagenesis, whether radiation or chemical, as GMOs? I see you have responded. If you can just do that verbally as well, please.
Thank you, Ben. And I will just repeat my input or what I was saying is that I did not make an attempt to define modified, but just to say that it is qualified in the way that the Act or the definition of the GMO is stated, and it's qualified by does not occur through naturally through mating or natural a combination or both. But then going to the actual question as to where do we place mutagenesis, I think I want to refer the query, the person who asked the query, to the Act, because the Act is then very specific in terms of what it excludes. There's another section that says, that indicates the exclusion of mutagenesis where GMOs are not used as parental or recipient organisms. So, and I suspect the drafters of the original Act were considering the history of safe use of these techniques, and therefore it was specifically excluded in the way that it was excluded at that point. Thank you, Ben.
Great, thanks very much. I will go to a next question now, the most recent one posted, which was really prompted by Alejandro's comment about the origin of the mutation and whether you can detect it or distinguish it from the original conventional variety. The question is, if a country cannot determine the modified versus the traditional developed products, how will it prevent imports from countries where GE is not regulated and therefore not labeled? This obviously has got a lot of interest happening around the world because this is the hot potato. Alejandro, your thoughts on this?
Yeah, I think that's a big question because you have the WTO norms. And in order to stop the commerce to trade, you have to accomplish for the WTO risk assessment. So what are you going to say? It's something that you cannot detect. It's something, so you cannot block the trade. So that's why I'm telling you, this is a technological default, because you cannot detect it and you cannot use it if you have norms against yourself. So from a technical point of view, you cannot stop trade because then you'll have a demand against you because you don't have the proof, the burden of proof. So you don't have burden of proof. So you'll be demanded by a country, and there is no way to solve that from a government point of view because you cannot establish a risk assessment of something that you cannot detect as different from a conventional product. So that's the problem in international trade. Now, I wanna say something else, and it is that in the past we have had products that comes from mutation that requires management in the field. So, for example, we had in Costa Rica Clearfield, which is a rice that resists a herbicide, and it comes back from a mutation. And what we did is to establish a management from the field in order to provide management and they work together with the Minister of Agriculture and Farmers. Now that's something that's completely possible. I mean, we have other norms. What we're saying is that not that we cannot manage traits that are specific and has to deal with a specific management, which is something possible, but that this is not a GMO from a technical point of view because from us, you cannot differentiate it, right? And if you cannot differentiate it, that's like going to the market and say you're selling water, and I'm asking you to provide a label when this water comes from rain or a river, right? And that's okay. But you can label river, but you don't have a way to know the origin, right? So that's the big problem would be in the future how to differentiate those in the market and how to provide. If you want to have a specific market, because that's also possible, to provide the way to provide that product in the market that wants that product, right? But from a technical point of view, you have an issue there because in the trade and at the WTO, you have to accomplish from a block of trade with the WTO rules. And in order to accomplish that, you need to get into a risk assessment of something that is the same and you cannot differentiate. So that's a challenge. I don't know how to solve that. That's a big issue.
Thanks very much, Alejandro. I think that was very clear and is going to be one of the challenges that has to be considered in a global system of trade using genome edited products. I wonder if I can follow up with Donald. Donald, you mentioned, you displayed in your slides that 25% of modified organisms applications to the am I regulated question are from universities. Now, I can understand that from an academic perspective and field trials. Are any of these progressing to commercialization? And if so, how are they moving along the value chain? Surely universities are not commercializing the products directly, but do you have any insight into that?
So I think that to date, there have been actually very few genome-edited products that are making it to the marketplace. One example, though, would be modified soybean oil through genome editing. And typically, these products would end up going through or being handled in a bit of a closed-loop production system because, again, with the concerns around what export markets and how they would be treated in that. The approach so far has been to try and control the production in a way to be able to keep track of material. But most of the applications that you will have seen through the MI regulated process are not yet products going headed directly for the marketplace. In some cases, they're even hypothetical because you didn't actually have to make the product to be able to go through the MI regulated exercise to get the decision on whether your product would be subject to regulation or not.
Right. Thanks very much. I'm going to refer to the question and answer, the question box. The question from an anonymous attendee is, are we over-regulating? Donald raised an interesting point where access to technology to market is lower. More SMMEs are active on a wider array of crops. The opposite also seems to exist, i.e. over-regulation stop local development. How many South African developed crops have been released under GM, not those events taking outside of South Africa and then locally released, but local GM events? Julian, if we can pose that one to you, that the question is really, do we have multinational GM products, or are there local developers as well that are developing and releasing genetically modified organisms? You're on mute.
Yeah, sorry, I was just muted. Yeah, Ben, unfortunately, the situation is that most of the developments, especially at a commercial release stage, is from multinational companies, where we've seen South African product or South African-based applicants coming into the system. There was still a very strong partnership with a multinational company. So that is unfortunately the situation, but I think it also does speak to the national system of innovation, because I think it would be an imbalance if we have this discussion around the regulatory system, and trying to pitch that as a constrictor or a constraint versus that prevents actually local guys coming into this area. But I think one really needs to also consider what is the national innovation system, what kind of support is there to ensure that these guys do come into the system. But regrettably or unfortunately, a lot of what we've seen is multinational companies. One just needs to bear in mind that the local regulatory system has a specific purpose, and we can't delineate that purpose from locally developed product from product coming from elsewhere. Thank you.
Excellent. I was going to follow up that with asking you, Julian, whether it's an indication of over-regulation or whether rather it's a local capacity issue that we're dealing with. Do you want to respond to that? I know I'm putting you on the spot there, and you may not have the answer.
No, as a regulator, I would not want to admit that we are over-regulating. Obviously, the client base would have a different opinion. But I've said it in my presentation, Ben, in that the regulatory system in South Africa, I still believe has evolved in line with our experiences and in line with what our domestic what our domestic realities are. So it's not an easy answer, but I think in how we've approached it up to now is that we've always been trying to accommodate areas where there is a lack of capacity. We've never lowered the requirement, but I mean there are other related processes where, as and when there's a problem, that we would assist the applicant with. But the system unfortunately needs to be. We can't have a system that is too fluid. But there should be ancillary processes to support applicants, and I believe that we make an attempt to provide that as and when required. Thank you.
Thanks, Julian. I note in a survey that the Biosafety South Africa platform put out that questioning whether the fact that the South African regulator has not yet decided to regulate genome-edited organisms has had a negative impact on the R&D bioinnovation program, and over 60% felt that at this stage there's been no real impact, but it will have in the foreseeable future if we do not resolve it. So essentially saying that we are gearing up, but we are not quite there yet. So good response, thank you. A more technical question, and then I'm going to go to the answers to the polls. So first, a technical question, the last one that's been posted for Alejandro. How does the inability to differentiate between the genome-edited plant and a conventional plant impact on plant breeders' rights and intellectual property? The issue associated with this, and perhaps you can touch on it as well, is people, for a product to be commercialized, there needs to be some way of identifying it. And so a commercial operator would not want their product to be indistinguishable from other conventional products. And so perhaps you can speak to this on the effect on plant breeders' rights and intellectual property.
Well, if you check the system right now, you have something that is called UPOF, and you have to accomplish for certain characteristic of the variety. So it has to be homogeneous, stable, and it has to have a different characteristic, right? But you're talking of a system that provides rights for the developer. So that system protects the developer, but also the user, because you're ending up with a variety that contains what it's supposed to have, right? And because genome editing is just another tool in breeding, then it's part of the breeding program. And it ends up with a variety that is covered under the umbrella of UPOF, right? It's under the umbrella of a variety. So from a right point of view, you'll end up with the rights of the plant breeder under the umbrella, under the development of a variety. Now, I'm probably more worried about the local developments in other areas, because right now, for example, I'm developing a yeast. It's right here. I don't know if you can see this. And this yeast is for producing biocombustible. And it tolerates salt because when you are producing biofuels, the yeast has to be more osmotolerant, and it has to be more strong. So what we're doing is that we're removing a gene that in the yeast, in the microorganisms that produces alcohol to make it stronger, right? So it's something simple, it has nothing to do. You can do it by other ways. You end up with the same thing, but it results in a very fast way. So we can use industrial yeast instead of laboratory yeast, and we can came back with a solution to an industry, right? That requires something now, right? And that is something that is a problem that is from now, that comes out from a university, and that's something that I think that we need to help for other innovations. In Argentina, 80% of the innovations from these techniques come from small companies. So yeah, I think that that's a major issue. It's not about the IP in terms of how to reference that IP, because you have it under the umbrella of variety, but how also to promote the new developments in the local products in other ways. Yeah, thank you.
Great, thanks very much. So I'm suggesting that we have a look at the responses to the various polls that we have, and then return to the Q&A. There still are a couple of questions that I do want to touch on, but perhaps we can have a look at the answers to the polls at this stage. There were 4 questions raised, and if we can please display them on the screen so that we can see what we have. There we have. So the first question, alignment of regulatory processes governing genome-edited products is essential to what? So there is a cross-the-board opinion, although the highest one is to promote sustainable agricultural research and innovation as the highest one at 18%. Then remove costly and non-science-based regulatory hurdles at 12%. And then at 6%, minimize trade impacts of genome-edited products and ensure accessibility to locally customized genome-edited product solutions at 4%. So that was the first question. Can we go on to the second one, please? This was a simple question. Should they be regulated differently? And as probably expected, the majority of stakeholders believe that 76%, the significant majority, that they should be, genome-edited products should be regulated differently. But there is still a minority, 24%, that believe genome-edited and genetically modified organisms need to be treated the same. Excellent. Can we go to the third question, which was, does genome editing have a role to play in improving the resilience and sustainability of farming in the food system? That seems to be an unequivocal yes, that there is a definite role for genome editing to play. Not surprising, but it is overwhelming. And then the last question is, who is responsible for communication that promotes acceptance of genome-edited products? Here we have, again, across the board responses. The lead one at 40% believe that genome-edited product innovators, in other words, the private sector and the developers, not only the private sector, but the developers, have a significant role to play. The next highest is regulatory agencies. That's Rufus and Julian and their teams have a responsibility to play in promoting acceptance of genome-edited products. And then, interestingly, academia still has an important role to play at 18% in promoting awareness and acceptance. It's obviously not clear what the other is. I would imagine the majority of the other means all of the above plus anyone else, but we don't have clarity on that. So some good and clear answers to some of the questions and across the board answers to a couple of them. If I then can go on to a question that's specifically on the South African regulatory system from Henny Groenewald. Dr. Jafta's interpretation of the scope of the GMO Act is 100% correct if and only if done from a process-based perspective. We have had the process versus product issue raised, I think, by Rufus. However, at least some formal legal interpretation has concluded that the Act is ambiguous in terms of its purported process or product basis. Does the Executive Council in South Africa consider addressing this legislative ambiguity before finalizing the genome edited guidelines? Julian, if you can have a stab at that, please.
Thank you, Ben, and thanks, Guinea, for the question. So my understanding is that we definitely have a product-based definition. If you carefully dissect the elements of that definition, to an extent, I agree with you that if you read different sections of the Act, that there is this issue of ambiguity, because the purpose of the Act definitely also refers to a process approach. How these, and should it be separated, or whether it is in fact a problem, I'm not 100% certain, because I mean, if one now gets into an argument of where the respective risks would reside, I can well appreciate that when the scope of the Act was written, that it obviously would have included the process approach as well. So I think that's definitely, it's always something that comes up, but I don't have a clear answer for Henny. But in terms of the amendment of the Act, it's obviously not a decision that we would be able to make. It's one of those things that we've always considered, because we've always considered the pros and cons of opening up the amendment of the Act, because I think the stakeholders must be aware when you start an amendment process, you will not be able to limit the amendment process just to that particular area that you now have an interest in. So it was the approach to try and work within the existing framework, because there might be other unintended effects, and the pun is intended, if you open up the whole Act for a review. But it is definitely something that we've considered and decided against at this point. Thank you, Ben.
Great. Thanks very much, Julian. I have another question for you, so please don't leave just yet, but we have another 8 minutes of Q&A, and then we will wrap up. The next question then is for Rufus from the chat box. The question reads, in Nigeria, for genome-edited products that are not considered GMOs and given biosafety authorization, are there other regulations that are applicable, or is the product essentially unregulated? This also refers to Donald's comment during his talk about the fact that all products are regulated. But Rufus, if you can talk to the Nigerian situation of unregulated genetically modified products.
The Nigerian process, as earlier on said, was the law which was amended in 2015 to include all edited products, states that all edited products will be regulated. In that regard, looking at the fact that with the definition of GMOs, when there are no novel genes in the final product, under the Catalina protocol cannot be considered to be genetically modified. In that respect, we had to do some guidelines to ensure there are some exceptions in doing a detailed regulation of the genetically modified edited products. So in that regard, we have the the three approach to it, the one that is edited without application, without the inclusion of any transgene or foreign material. In that case, we will not do detailed risk assessment, but we only give a clearance that it does not fall within GMO definition. Then the one that is transgene is being used, but at the final product, the gene of insert does not appear in the final product. In that case, we also give some level of exemption from detailed review process.
We seem to be having some connection problems. So in the absence of a clear connection, can I then mute that? And unfortunately, not a full response from Rufus, because the connection then prevented us getting the clarity. But perhaps, Donald, you can just touch sides on what you meant by unregulated products. They may not be regulated under the GMO, but you're saying that they are still regulated. Perhaps you can just give some clarity and perhaps an example of that other regulation.
So if we think about, the conventional regulatory system, and in many countries, we would have requirements, for example, for variety registration. So that, new crop varieties that are developed by breeders have to demonstrate not only, DUS, distinctness, uniformity, and stability, but they also have to demonstrate value for cultivation and use. So they have to be both safe and efficacious. We have regulations that cover food production and processing and manufacturing in all manner. We've got regulations that cover the grain trade, and that in terms of setting standards, and also setting standards for seed quality, seed certification, and that. So we've got this whole array of different regulations that can be used. And as I said, some countries have chosen not to create a special GMO Act, but rather to say, well, we already have a Seeds Act that controls the introduction into the environment of seed. Well, that means any kind of seed. It means a biotech seed. It means a conventional seed. It means seed. And so we could modify that act if we wanted to be able to deal with seeds that are products of biotechnology, and for that matter, products of genome editing or genetic engineering, or even mutation breeding. We go back and look at Alejandro's map. There was one country that was colored blue, which was totally out of step with all of the other countries in the world, and that was Canada, which has been regulating based on a product approach since 1993, and dealing with how do we define what a novel trait is, and what a novel plant is, and what a novel food is. And so developers in Canada have been having that discussion with regulators for the past 30 years on, is my new thing novel or not? Which is the same kind of discussion that many countries are now wanting to engage in terms of a pre-regulatory discussion around a genome-edited product, and does it contain a transgene or not, or is it going to be subject to regulation or not?
Great, thanks very much. Julian, I said I would come back to you. This is not a trick question, but can you respond to how far is South Africa from allowing gene-edited crops to be planted? Are we looking at months or years?
Thank you, Ben. Sorry, I just need to do something here. Okay, good. Given the long, extended time that Council as well as the AC has deliberated upon this matter, Ben, and also mindful that answer, whether it is an answer that people are not necessarily happy with, but an answer should be forthcoming from the Executive Council. We have had extensive discussion at the last meeting at the start of May. The next meeting would be in another 2 months' time, but Council had agreed that we will attend to the matter within the intercessional period. So there might be an answer before the 2 months expire, before July or end of July. But that would mean that there's a consensus. I'm assuming there's a consensus decision, and then there's a need for this to be presented to the minister. I can see another 2 months for such a process to be concluded. So if I can give a very conservative answer and not speaking on behalf of the council, I would give it another 5 months before we have before we are in a position to give a clear pronouncement. That's just how, if an educated estimate that I can give at this point. Thank you.
Thank you, Julian. Although, to be honest, I was expecting your answer to be the short, sharp one, which is to say that genome-edited products will be considered as GMOs at this stage, so they can be considered straight away. What Julian is referring to is the consideration of treating genome-edited products differently in some manner, which is as yet undecided. But genome-edited products, please correct me if I'm wrong, can be considered for release into the South African environment, but it will be under the current GMO regulations.
Yes. But that was my point. I did say in my presentation, and apologies if it didn't come out clearly, I did say it's considered under the GMO Act. However, the how, because we can say, yes, you have to submit an application, but we want people obviously to also be guided. And this is why the approval of the framework is important. Sorry, Ben, it's been a long day. I didn't get you clearly. Thank you.
Great. Thank you. Folks, we have now run out of time, and I need to wrap this up. I want to thank the 4 speakers very much for their input, for their presentations, and apologies to Rufus. We had to cut you off in your last question because the connection then was not adequate to get your response. But what I think we are clearly highlighting is the importance of this type of engagement, not only with direct stakeholders, but it needs to expand to all stakeholders and to the public and society in general. This is a new technology. As Donald put it, there's been a lot of, there is no evidence of additional risks, and there's no such thing as unregulated products entering the market. And what we need to recognize, as he said as well, time is short. If we want to take advantage of these technologies, we do need to act, not necessarily in the introduction of the capabilities to produce, but at least on the regulation that can consider them. Some really interesting talks, really appreciate it. And as I said, highlighting the need for further engagement. Thanks very much for your time. And I will hand over to Chantel then for the closing statement.
Thank you, Ben. In closing, I would just like to thank all the speakers again for their valuable contributions, especially those of you that had an early start to your day today. You know you are. Thank you, Ben, for officiating today's discussions, and also all of you, our audience, who joined in and participated in today's webinar. Of course, the conversation does not end here, so we encourage you to tune in again next month on the 22nd of June, when we'll be hosting a second event in the series that will focus on how genome editing technologies can support opportunities in agricultural innovation. So until then, we wish you well and enjoy the rest of your Tuesday.
Thank you.